Hi, my name is Robin Cooper, and I'll be giving you a presentation on recent journal articles and some related topics on the effects of deep tissue injury in muscle and on the healing processes that occur. I'm trying this approach where I'll be mentioning what slide I'm on with the PowerPoint, and then you can watch the YouTube video at the same time that goes along with this. The next approach will be the two screens together recorded as one movie, and we'll see how that works as well. Alright, so that was slide 1, and slide 2, we see here the human body, the skeleton, and the muscle that covers the skeleton. So one type of deep tissue injury can occur when a object, or maybe from a fall, hits the muscle and then pushes against the bone internally. So you could have an external wound as well as an internal wound from compression against the bony processes inside the body on the muscle. You can also have injuries such as this pull-tear injury from running for a long period of time, for example, or maybe overdoing it and you stretch your muscle and you tear it or damage it internally. And you can have a bruise such as this approach. You can see it's blue, but the skin's not broken in this case. And in time, both these types of wounds can actually break the surface and end up with an external wound. So this deep pressure injuries, such as internally there or from the object hitting your muscle, can make a wound on the inside and work its way outward. Or, like we mentioned, if it's an external object that hit the muscle, then it could be an outside wound initially, but also have severe tissue damage on the inside. Both of these types of wound can result in further muscle and tissue damage. So this is what's referred to as possible deep tissue injury. And this is, I'm going on to slide 3 now. Here we have deep tissue injury where it's more than just the acute damage on the tissue, such as the breaking of the skin from hitting the object. But you see in the slide on number 3 is that the surface of the skin, it might show a very small bruise on the surface, but underneath there's a larger tissue damage. So it's kind of like the expression of the tip of the iceberg. You see the tip, but you don't see what's underneath it. And this type of injury is one that can lead to what's called a pressure ulcer, or look like a pressure ulcer in a stage 3 or stage 4, where the skin will be broken and there'll be deep tissue underneath it that is injured. So on slide 4 now, you can see the causes of the deep tissue injury, that's the DTI development, and they're multifactorial. They're a complex series of events, such as, if we hit the arrow keys as we're going through this, the bony muscle interface deformation, like we mentioned, between the bone and the muscle, ischemia, ischemic reperfusion injury, that's after the blood stopped and then it's reperfused again. That can actually cause injury to the tissue as well. And impaired lymph drainage, the drainage of the lymph fluid out. If there's a compression and the lymph can't drain, this can build up the lymph and actually lead to more tissue damage. Any alteration in the interstitial fluid flow, such as going into the lymph, can also cause more tissue damage. So this is more of a compression problem where the fluids, blood, and the interstitium cannot flow properly. And as we mentioned, this edema can be formed from damage of the capillary beds and the capillary bed permeability increasing, and then fluid leaking out of the circulatory system into the interstitium. That then increases the swelling in that area if it can't drain. So this is edema. And then from these kind of processes, you can have inflammatory changes that occur in the body's reactions to this. And then this can trigger a process called apoptosis, which is regulated cell death. All right. If we go to slide 5 now, how to prevent further muscle or tissue damage as a result from the primary injury. And we see that, 1, you'd want to prevent the skin breakdown from an open wound, but also increase the blood flow so you could get rid of any waste products that are coming out of the damaged tissue, the muscle, and also keep that interstitium flowing. So some people think that applying heat might be good. Others think applying cold to keep the swelling down might be an approach. Also, medications are of interest here because they can be anti-inflammatory and help in blocking prostaglandins and more tissue damage that could occur from that. Also, steroids such as glucocorticoids help reduce the swelling. The current recommendations, though, that are to be followed for standard care is to treat them as their pressure ulcers. And usually pressure ulcers then are treated such as repositioning a patient, a client, or putting maybe one of those air mattresses on, a waffle mattress, and keeping the skin from breaking down any further, topical dressing applications, and of course nutritional support to promote healing. But this time there are no new treatments that have been standardized for treating deep tissue injury. Luckily, there was a fellow over here in Lexington, Kentucky, Jeremy Honaker, and Michael Forston, the first two authors on this paper on slide 6 that you can see. They are working on this project and actually doing a comparative study to find out if this new type of treatment, which they're starting to use, is actually helpful or not. And this is with low-frequency ultrasound on the healing of deep tissue injury. And they just published this paper in International Wound Journal, so it's a pre-press article. I'll put it up online as well with these PowerPoint in this video. Basically, what they were after was to determine if the ultrasound therapy would be beneficial or harmful. And ultrasound therapy in this case is delivery of ultrasound at low intensity and a low frequency of 40 kilohertz. There's no contact with this low frequency ultrasound, so it's a mist. It's, as you'll see in a minute, it's a saline mist. There's no thermal effects with this healing, and that could alter conditions if you had thermal as well as this ultrasound. That's another variable. So what they use is this low frequency ultrasound wave, and it penetrates deeper into the tissue than, say, long wavelengths ultrasound. So let me show you in the next slide, on slide number 8, it is. You can see the instrument that's used. It's a saline container and then this ultrasound device that delivers ultrasound. And the saline is what carries the ultrasound onto the client. And if you hit the arrow key, you see a little piece of tissue here with some rain droplets. If you click on it, they might move up and down. And that saline then penetrates deep into the tissue. And it's also thought if there happens to be bacterial cells there, that this ultrasound frequency can actually break up the bacteria the way it's the frequency that they're giving. So it's kind of interesting. I didn't find much of the primary literature of that being tested inside of tissue in an animal, but the publisher of this device actually states this fact. For this study, they measured 3 characteristics of the suspected deep tissue injury, so that's the DTI there. We're on slide number 9 now. Were used to identify their severity. They used the total surface area, TSA, skin integrity, and the wound color, or tissue assessment. And they came up with this index, which Jeremy Honaker put together, and you can see it over here in the bottom. It's the total surface area, and depending on the surface area of the wound, and plus, they would then use it times skin integrity, and you can see there it's intact or broken skin, and then they would plus that to the wound color or the tissue assessment, such as does it look bruised, is there necrotic tissue or not. They could then add these numbers together and get a total score of that wound. So some kind of assessment that they could give when they first see the wound, and then when a patient's discharged from the hospital, redo the same assessment and see if giving the therapy or not giving the therapy, if there was some benefit to it. The take-home points of their study is that the total difference noted between these groups showed a significant decrease in the wound severity. So their score, the total score, was lower by 2.52 on this 3 to 18 scale. So with the NLFU in the control group, that was that non-contact low-frequency ultrasound versus the control group not giving it at all. they actually had an improvement in the ones that had that treatment of the ultrasound therapy. So they're suggesting that those findings were actually beneficial for treating the suspected deep tissue injury, and these were the first attempts to actually validate, to quantify the treatment versus control group. So it's actually a good study that came out. I didn't go into all the details of their exclusion and possibilities in why they excluded different patients, such as severe open wounds and things like that. But you can see that in the primary article. What's more interesting, I think, to the biology audience here is the mechanisms of how this is thought to work. So I'll focus on some of that. A low-frequency ultrasound has been shown to upregulate endothelial nitric oxide synthase, NOS, secondary to, say, the ultrasound's stress by cavitation. That's this the sound going in between the tissue, and microstreaming of that fluid with the ultrasound into the tissue. So what they found is with this NOS itself, that can increase blood flow. So improving blood flow by just stimulating NOS, for example. Decrease muscular reperfusion edema. Well, if you increase blood flow, you increase the lymphatic flow, you can increase that fluid to escape out of that damaged tissue. Also that the inflammatory response was decreased. So leukocytes going out of the capillary bed and stimulating more of an immune response was decreased. Free radical induction and leukocyte chemotaxis was altered, and decrease in the nitrogen and oxygen delivery for free radical formation. One of the factors that is thought to really contribute is this upregulation of vascular endothelial growth factor. So that could increase the capillary bed and actually help to promote circulation to damaged tissue, especially if other capillary beds were closed off. Also, this decrease in the tissue necrotic factor, or TNF alpha, and interleukin-1, these can help decrease the cytokine productions and then decrease the immune response as well. So all these are really thought to be connected in with the NOS, and NOS is upregulated by this ultrasound treatment. So local responses that can actually help in this tissue repair. And that might be some of the underlying mechanisms of this treatment. So what's next? This is on slide 12 now. What should be done next? Well, Honaker et al. Really feel that the available literature does suggest that this ultrasound is capable of stimulating cellular activity and mechanical energy in the absence of adding these extra factors such as ATP and cytokines, other enzymes. So, what kind of further treatments should be done, and to what kind of wounds? I think that's now where they're emphasizing. Maybe certain wounds this won't be so beneficial, and others it would be. So they need to find out more about this type of therapy and see if it's warranted for particular types of wounds that they can define. If we go on to slide 13 now, I put in this slide from reading their work and what we can gather on this tissue repair. I would think it would be really interesting if you could measure blood flow with Doppler techniques, and you can actually see while you're giving ultrasound that you see an increase in blood flow and how extensive that increased blood flow might be. It'd be interesting, maybe in animal studies, if you could measure glucose uptake, either radio-labeled or and other approaches that you could actually see if the tissue's being uptaken by increase in this metabolism in injured cells, for example. If you're causing repair, maybe the repair processes are going on quicker, enabling cells where this therapy is happening. Tissue perfusion, this is something of interest. It's if you have a deep tissue injury and you've damaged a lot of muscle, a lot of the cellular contents will now go into the interstitium and then into the lymph and back into the blood. So you can actually load up your blood, very high levels of protein content depending on the amount of tissue damage. This can also cause renal failure because you can clog up your glomerulus with high protein from so much tissue damage. So one has to watch out for kidney failure in such cases if it was a massive amount of tissue damage. So possibly, I'd be interested to know if tissue perfusion, if you put in hypodermic needles, for example, or maybe larger needles into this area, could you perfuse a large area and actually wash out the cellular contents before it gets into the lymph and back into the circulation in the blood, and then probably avoid some of the potential problems that could happen with kidney failure. And I'm interested to know if when you damage the tissue, the potassium levels, because potassium is higher inside the cells, that potassium would spill out, and that potassium spilling out of the damaged cells could then cause further injury in the neighboring cells by depolarizing them and causing those cells to be leaky. So I'm wondering if this high level of potassium, potentially high level of potassium, could be a contributing factor to the spread of deep tissue injury. This would be an interesting subject to possibly test in non-human studies if one injected certain levels of potassium chloride, for example, into muscle, in rat muscle, that how much damage would that occur and how wide could that damage be from the zone of injury. And then if that would correlate, the levels would correlate to what one would measure in such tissue injury. This potassium idea is not totally new in the sense that in the 1930s, T.P. Fang, he noticed that potassium can be very toxic to cells. And I'm on slide 14 now, where I take this quote from his work, crushed muscle with the concentration of potassium to diffuse cannot avoid being toxic. In view of the fact that Sidney Ringer, when he was developing his Ringer solution, contained only 10 times the normal concentration of potassium, and it was already could be toxic. When he was making this Ringer solution, he would increase the potassium, Sidney Ringer, and notice that could damage the cells. So I think the potassium has a potential of adding to the problem, and maybe the perfusion of the tissue would be one way to decrease its potassium levels. So this could be an interesting way to maybe remove excess potassium by perfusing those areas. Or even if you had a muscle tear or sprain, a severe tissue damage, maybe the perfusion of that area would also be beneficial to decreasing excess potassium. All right, and if we go on to slide 15, this is another paper by Solis et al. In 2012, just came out. And this is on the distribution of internal strains around bony prominences in pigs, where they actually, you can see in the bottom slide there, the figure, they have an anesthetized pig and this little pressure indenter that goes on the bottom of the pig. And they could actually relate to the pressure and the vectors of pressure in different regions on the muscle that's going to be pushed against the bone, this bony protuberance in the hind of the pig. And what they were really aiming at in the next study that's a sequel to this study is that if they stimulated the muscle with electrical stimulation, so this is another procedure that's being tested now, electrical stimulation, could they enhance muscle repair by increasing blood flow and muscle contraction? So they did some initial studies with this pig like this, and they could then look at the injuries. If we look on slide 16, they could then with the MRI actually look at the injury that is given when they push that protuberance indenter against the back end of the pig. And they could actually quantify, if we go to slide 17, they could quantify the amount of tissue depending on the pressure that they would give. And with this, they could do then measurements and see about how much pressure it would take to give so much injury. If we look at the top of the slide on 17, you can see it says 25%. That's 25% body weight, and then measuring this deep tissue injury. And then over on the right-hand side, they could then measure 50% body weight and then see how much tissue injury they were given. All right, so it's kind of interesting to then do these measurements on these pigs. And if we look at slide 18, the take-home from the slide is that they found that those loads that they were pushing onto the pig were very similar to the loads that a human would then experience with injury in the muscle. Then they wanted to compare what is referred to as electrical stimulation, and then seeing on how well that would cause healing of the muscle. They found in this study if they loaded the muscles, like stimulate them or have them loaded where they're not relaxed, then they could actually have less injury on the muscle than if it was an unloaded muscle. So if the muscle's not contracted, then it would actually cause more injury. Alright, given the substantial physical and psychological economic costs with these deep tissue injuries in humans, this is a really neat study to find out if there might be a way of preventing or enhancing the healing of these deep tissue injuries. As we see, as I'm going through the arrow keys here, I'm down to the electrical stimulation for prevention of pressure ulcers. Was first proposed back in the 1989 and late 80s. And this was thought to basically help to improve blood flow. From these various studies that were put out since the 80s, late 80s, it's basically the stimulation could help in having the muscles contract and increase blood flow. It's no different than really if you hit yourself on the bottom, for example, on your leg, you wouldn't feel comfortable and you would fidget, move around, and increase the blood flow to that muscle where you wouldn't be pushing on that injured side all the time. But if you had a back injury and you had spinal injury, you wouldn't be able to move around so easily. So possibly by using electrical stimulation would be an approach for somebody that might have back injury that can't move around themselves to increase the blood flow in these muscles from being contracting. So in another study, a follow-up study right after this last one, they actually used humans and they looked at the pressure that would be given, say, sitting in a chair, and then they set up, and that's a slide on number 19, where you can see in the middle the blue figures there. The white that's in there, that's the pressure that's being measured. And then they use this MRI setup where they could simulate like a chair against the bottom of this person, and then detect the pressures that were given onto the internal structures. And in the very bottom, you can see what that pressure is pushing on the bony protuberances inside the bottom, and it's pushing against that major muscle, gluteus maximus muscle, on the bottom of the patient. So what they wanted to do is then compare electrical stimulation and look at those pressure differences. If you go to slide 20, you can see now the first picture on the left is at rest and on the bottom parts of the bottom. If you look at the middle row, the IES is labeled at the top. They would stimulate one side, like on the left buttock, or in C, stimulate the right side buttock, or in G at the very bottom, they stimulated both sides of the buttocks. And you can see on the right-hand panel then, where the dark red is, that's where the pressure increased from their stimulation. And at the very bottom, you could see that also, bottom right, at that red, they got pressure increases. And then in some areas, no changes at all. Well, the take-home from the study was that they showed that there was an increase in blood flow and oxygenation to the gluteus maximus from the stimulation paradigm. And on slide 21, you can see from these bullet points here, when the muscle was compressed, the inflow and the outflow of the blood was actually compromised. It could occlude the area, and this ischemia could occur. And that prevents the exchange of the oxygen. But with the muscles contracting off and on with the stimulation, that promoted blood flow. Part of this increased blood flow is a bit of a paradox in the literature, because reperfusion of an injured site can actually cause more injury. Depending on how long it hasn't received oxygen or blood flow, it can actually enhance the injury even more. So it's kind of interesting. It's a similar kind of case in the brain as well with strokes. Well, here they think that if the muscle is exercised, the idea behind this electrical stimulation, nitrous oxide is produced, which can inhibit the mitochondrial respiration and reduce the consumption of oxygen. So if you stimulate the muscle, you stimulate nitrous oxide, which reduces mitochondrial respiration, decreases oxygen consumption, and then you have more oxygen around. And then it won't be such a problem for that tissue immediately after muscle contraction. And because of this long-term effect of active contractions of stimulating the muscle with electrical stimulation, then there should be lower oxygen demand, making the muscle more resistant to the ischemia that occurs. So this is the main point behind it. That's why I bulleted in red on the slide as you go through the arrow keys. And then the last point was that they also found out that electrical stimulation promoted better comfort to the patients that were getting it when they stimulated these muscles versus just not getting it all and sitting or laying still on the bed. So the comfort level was increased by this electrical stimulation of these muscles. In my view, kind of wrapping these studies up in this concept of deep tissue injury, I think it would be interesting to try both of them together. Why not try the ultrasound therapy and the electrical stimulation and see what kind of effects? Maybe get a synergistic effect in a much better healing process. Also, maybe assess still this potassium spillage idea from damaged tissue and see if washing out the toxin of potassium would prevent further damage and maybe promote a faster recovery and healing process. I think that would be quite interesting. And that's about where I am there, and I think we could really have some neat discussions about this concept and see what you all think.